Thank you. Thank you very much, Simon, for the introduction. Thank you for being there and for inviting me for this seminar. So today I will not talk too much about phage plasmids, but I will talk about phages and plasmids. And the focus of my talk will be on how this network of interacting mobile genetic elements actually shape bacterial evolution. So for this, I will have to introduce four different agents. And it's the interactions between these different agents within the cell and the genome, and that will shape the horizontal gene transfer and the evolution of bacterial gene repertoires. So the first one, of course, is the bacterium. Here I'm presenting you a nice picture of E. coli, the superstar of microbiology. And E. coli, it's an interesting characteristic, which is very plastic in terms of phenotypes and in terms of genomes, as we shall see. And some of these strains of E. coli are extremely pathogenic. Some it only requires an inoculum of 10 to 100 cells to make you sick with 50% probability. Whereas other strains are much less pathogenic. Inoculums of 10 to 8, 10 to the 10 are required to make a healthy person sick with 50% probability. And of course, most E. coli are actually commensal. They're not pathogenic at all for us. And it is a very important question, not just from a theoretical point of view, but also from a clinical point of view. What makes the difference between two strains of the same species? One of them it may be extremely pathogenic, and the other may be commensal. And this is particularly important because each and almost every one of us listening to this talk has about 1 trillion E. coli in the gut. And it's important to know exactly what are we dealing with here. So E. coli, like many other bacteria, has an extremely plastic genome. This is an analysis we did a couple of years ago. We analyzed 1,300 natural isolates of E. coli, mostly commensals, but also a few clinical isolates. We sequence all this, and the average genome of E. coli in our data set has about 4,600 genes. But within this, there's less than half of the genes, about 2,200, which are core genes. That is, that we can find in more than 99% of the strains of the species. And in contrast, there is 15 times more genes in the pangenome of E. coli, in the set of all gene families that we can find in the species, at least in our sample, relative to the average size of an E. coli genome. So the diversity of gene repertoires is huge, even at the level of a species. And we can see this just by looking at the number of genes we can find in the genome. This is protein coding genes. The smallest E. coli genomes have about 4,000 genes. The largest one have almost 6,000. So this is a huge difference in terms of gene repertoires in size. And this is what I'm representing here in the gray distribution. Now, the red distribution is the same thing, except that from the genomes of E. coli, we have removed two mobile genetic elements which we can identify relatively easily, which are plasmids and phages. And you see two things in this distribution. First, the average size of the genome decreased by about 10%. So these elements do take up a significant fraction of the genome. But more interestingly, the coefficient of variation of this distribution decreases by a factor of 2. That is, these mobile genetic elements, and there are many others, but just phages and plasmids are enough to make a significant difference in size of the genome, and more especially, they have a huge impact in the generation of variability in the short term in terms of the gene repertoires of the strains. Now, this means that I have now to present phages and plasmids. You've already had some talks about it. Phages are bacterial viruses. In many environments, they outnumber bacteria. They have a huge impact on the planet and bacterial communities. According to certain studies, they may kill up to 50% of the bacteria in the community per day. And some phages are temperate. This means that they have the ability to integrate the genome, and as they integrate the bacterial genome, they effectively become part of the bacterial genome, and they can express traits that are adapted for the bacteria. They don't do this because they're particularly nice. It's just that the temperate prophage, when it expresses traits that increase the fitness of the bacteria, it actually is increasing its own fitness, because every time the bacteria is replicating, the phage is replicating with it. Prophages are more than a third of E. coli's pangenome, and they do have a lot of genetic variation to the species. Now, plasmids are the second type of mobile genetic element I will talk about today, the third agent in my story, and they can have very diverse sizes. The smallest plasmids have no single gene protein coding gene. The largest plasmids have sometimes more than 1,000 and up to more than 2,000 genes. So the acquisition of one single mobile genetic element can increase the size of a genome by a very significant fraction. And plasmids are known to evolve extremely fast, to be able to acquire genes at high rates. And this is probably what makes them very successful at driving the spread of antibiotic resistance genes, virulence, symbiotic traits in rhizodia, and so on. So plasmids tend to transfer, not only, but most of them tend to transfer by conjugation, which is basically a mechanism involving a type 4 secretion system that connects to cells and transfers DNA, single-stranded DNA, from one cell into the other. So what we've been trying to understand is whether the types of mobile genetic elements actually matter in terms of the flow of genes in communities and in terms of the traits that are transferred between cells. And to analyze this question in detail, we have been studying in great deal of detail Klebsiella pneumonia, which is an enterobacteria, and for which we have now thousands of genomes. So Klebsiella pneumonia has an interesting trait, which is it has the cell is surrounded by a polysaccharide layer, which is a capsule, which can be extremely thick. You can see it here in my picture. It's the yellowish thing around the cell. So it can occupy a volume that it's larger than the cell itself. The capsule can be a barrier to mobile genetic elements because it's a very thick layer at the outside of the cell, but actually it also evolves by horizontal gene transfer. And what I'm representing here, it's a data set of about 4,000 genes of Klebsiella, for which we identified the core genome, made a phylogenetic analysis, and then identified for each genome the serotype. So each color represents a different serotype. There are many serotypes. You know, more than 100 different serotypes in the species. And they're not monophyletic, so sometimes you'll find the same serotype in different parts of the tree. Each strain in general has only one serotype, which is a very specific genetic locus in the genome, but keeps being constantly gained and lost and exchanged, and this therefore requires horizontal transfer. What we've been trying to understand is how the interplay takes place between the cell and its capsule and the mobile genetic elements, knowing that horizontal gene transfer may be hindered by the existence of a capsule, but on the other hand, horizontal gene transfer is necessary for the variation of the capsule. So the first thing we did was to try to understand if the serotypes somehow constrain gene flow. And to do this, we take our big tree and we identify cases of recent horizontal gene transfer. And we look at gene families like here, the white stars and the black stars, look at genes of the same family that recently been acquired in two different parts of the tree. And we just analyze if these co-occurrences of transfer of the same gene take place preferentially between bacteria that have the same serotype or if they're just random. What we observe is what I'm representing here in this graph is that there is a significant bias. There are twice more frequent transfers between bacteria that have the same serotype than what we'd expect if it was random. Now, you have all these genomes, you have all these transfers, and you can remove the transfers due to plasmids. And what you observe is that the bias increases. And you can remove the transfers due to phages, and what you observe is that the bias decreases. What this suggests is that this intra-serotype gene flow preferentially driven by phages. And to test this idea, we did what we call a phage-bacteria interaction matrix. So we took a large panel of strains of Klebsiella, we induced the prophages of the strains, we recovered the phages, the lysate, and then we challenge other strains with these phages and see if they manage to infect. When it's white, it doesn't infect, and actually phages tend to be quite specific of a few strains. So in most cases, we have no infection. But in some cases, we have infections. In yellowish cells, it's infections of phages that come from one bacteria and infect the bacteria with a different serotype. And in blue, it's when the phages from one bacteria infect bacteria from the same serotype. And what we observe is that in 33% of the cases where we are exploring interactions between bacteria of the same serotype, the phage manages to infect. Whereas when bacteria have different serotypes, this only takes place in less than 4% of the cases. So what this really shows is that the fact that the bacteria have a specific serotype will constrain gene flow in the community such that gene flow will preferentially take place between bacteria that have a given serotype. To put it more broadly, I won't have the time to go into details, but the paper is published If the bacteria lacks a capsule, the phages, because they have co-evolved to adsorb to the capsule, will be less efficient. And so bacteria lacking a capsule will be somewhat sexually isolated from the point of view of phage-driven horizontal gene transfer. And so this raises a question of what happens to these bacteria that have lost the capsule. They're now immune to phages. That would be great, but one of the costs that they could have to pay would be the fact that they would not have horizontal gene transfer because they are not infected by phages. We can go back again to our big tree and identify cases where the capsule was recently lost. So here the yellowish circles represent strains, extant strains that have a given serotype. They all have the same serotype. And here you see in the terminal branch, we have the loss of the ability to produce a capsule. So now we can identify all the horizontal gene transfer that took place in terminal branches, in the branches of bacteria that are capsulated, bacteria that are not capsulated. And we see that the bacteria that are not capsulated actually have more horizontal gene transfer than the bacteria that are capsulated. Since this is probably not due to phages, because we saw bacteria that lack capsules tend to be resistant to phages, then what's left? It's conjugation. And what it suggests is that conjugation has a very different effect on gene flow than phages. So to test for this, we made a large analysis where we took 3 different strains, 4 different serotypes, and we created 3 groups of strains where in each group we have strains with 1 of 4 different serotypes and 1 variant which has no capsule whatsoever. We have a large number of plasmids, and then we can conjugate all the pairwise, compare all the possible pairs between our strains, and we draw sort of a map of the frequency of conjugation depending on the serotypes. And what we observe is that here the delta, that is the strains that do not have a capsule, do indeed conjugate a lot among themselves and conjugate significantly with the others. In contrast, there isn't a bias towards high frequency of conjugation between bacteria with the same serotypes. So mobile genetic elements do matter. Conjugation patterns and the patterns of transfer driven by phages are very different. Why is it that conjugative elements seem to conjugate more easily between bacteria that lack a capsule? We think it's because of the volume of the cell. I showed you that the capsule takes a lot of place and probably hinders the contact of the conjugative pillars between the 2 cells. And this is sort of a demonstration of that. We have the 3 strains that we analyzed in red, in blue, and in yellow. And in each strain, we have the strain lacking the capsule, the delta, and the different serotypes that we've tested. Some produce thicker capsules than others. And what you observe is a nice negative correlation between the volume that we observed in the cell with the capsule and the frequency of conjugation. So it does seem that the capsule, because it's so thick, will hinder conjugation in many cases. To sum up, the existence of the different types of mobile genetic elements with different mechanisms of transfer will indeed have a very strong impact on gene flow that we observe. In one case, phage-driven gene flow will be very frequent within the strains and rare between strains, even rarer with strains that lack a capsule. In contrast, conjugation will be very frequent between bacteria that lack a capsule, but frequent across the entire species. So different elements will have different impacts in gene flow because of the way they interact with the cell envelope. Now, once the bacteria acquires the mobile genetic element, the phage injects its DNA or the plasmid gets conjugated into the cell, there is still an extra layer that the mobile genetic element has to pass before it becomes integrated in the genome, or it becomes functional in the case of virulent phages. And this is the immune systems that exist in the bacteria. Now, many have been discovered in the last couple of years, but the most frequent of them is restriction and modification. And about 80% of the bacteria carry restriction modification systems. And the reason for this is that even though phages and plasmids can eventually bring to the bacteria adaptive traits, they are also costly. What I'm presenting in this figure, it's a picture of E. coli once it has been infected by a virulent phage and after the lysis of the phage and liberation of the viral particles. And what this reminds us is that even though these mobile elements can indeed be helpful for the bacteria, can bring adaptive traits, and they can also be extremely costly. And in the case of phages, they can simply be deadly. So bacteria have developed a very large number of defense systems to protect themselves from the elements, and the most frequent and the best known one is restriction modification. In this case, there are two functions, a function of modification of DNA, typically methylation, and the function of recognition of self from non-self, that is recognition of these methylation patterns, that will direct an endonuclease towards doing either nothing or destroying incoming DNA. Restriction modifications should have an impact on gene flow, but we wanted to understand how strong was the impact, and if actually we could observe it by looking at the genomes. In terms of two things, gene gain, so gain of new genes by horizontal gene transfer, and just in terms of allelic recombination, so homologous recombination in core genes. To do this, we did a simple analysis. This has been published a couple of years ago. We took about 80 species, and within each species, we looked at strains that were compatible from the point of view of restriction and modification. That is, two strains that have the same type of restriction modification system, such that if the plasmid goes from one strain into the other, it has the same methylation patterns, and so it should pass freely without having an immune barrier. And we can compare these pairs with pairs of bacteria that have different restriction modification systems, and you would expect an effect of restriction that will hamper the transfer of the mobile element. So when the calculations were done, we did these on 79 species. We had fewer genomes at the time that we have now today, but still the effect was very significant. There is significantly more horizontal gene transfer, twice as much, between strains which are compatible than compared to strains which are incompatible from the point of view of restriction and modification. And there is 8 times more homologous recombination. So this second layer, the second barrier to transfer, which is the set of immune systems of bacteria, can indeed have a strong impact on gene flow in the communities. But at this stage, we thought we had things more or less figured it out. Of course, we don't know the details, but we thought we had figured it out, the overall set of determinants of horizontal gene transfer. First, it comes contact. The cells must be in direct contact for conjugation, or the phage must be in direct contact with the cell to infect, which means that horizontal gene transfer will tend to be more frequent within the same environment. And then there is the establishment of infection. I just showed you the case of the capsule in different bacteria. It can be an effect of the capsule or other outer membrane associated structures. And finally, there's a layer of immunity that is important for the mobile elements to pass, to be able to infect and eventually be part of the bacterial genome and bring it new traits. And finally, of course, there is the question of the function of these genes and how adaptive they are for the bacteria. So the establishment of the genes in the long term will depend on the effect that these genes have on the bacterial fitness. And so this seemed to be a sort of scheme of things, and the pairwise interaction between the bacteria and the mobile genetic element would seem to be key to understand the patterns of horizontal gene transfer. But in the last couple of years, a number of discoveries changed significantly the way we think about these things. And in a way, it rendered this picture more complex, but also quite more interesting. Because one comes to realize that we must go beyond the pairwise interaction between the mobile element and the cell if we want to understand what's really going on. And the reason is the fourth agent I want to present today, and these are the hyperparasites. Now, I'm calling them hyperparasites, but in so many cases, we don't actually know if they have a significant cost on the element they're parasitizing. But let me go into the detail. We now know that many plasmids, for instance, are not able to conjugate autonomously from one cell into the other using a type IV substitution system that they would encode, but instead, they will hijack the conjugation apparatus of other cells, of other conjugative systems. So you'll have mobilizable plasmids that will hijack the conjugative apparatus of a conjugative plasmid to move between cells. And they're called mobilizable plasmids. We know they exist for a long time, but we now are actually able to quantify this. And we know that the vast majority of plasmids are actually not conjugative, and most of them are mobilized. Recently, we also came up to the ability to quantify the existence of an element which has also been known for decades, but until 2 years ago, only a handful of elements were known. And these are phage satellites. So phage satellites are mobile genetic elements, which are not phages at all, but are able to hijack viral particles to move between cells. So as the phage is producing the viral particles where it will package its DNA, MGEs, these phage satellites are able to still package viral particles from the phages and package their own DNA so that they can transfer between cells. So in both cases, we have an aproparasite because the phage satellite will be a parasite of the phage, which is in itself a parasite of the bacteria. The bacteria, of course, can also be a parasite of an eukaryote, but let's stay simple for the moment. Now, what changes fundamentally now is that we have a sort of a hierarchy of functional dependencies in the cell. We have the mobilizable elements that will depend on conjugative plasmids or in phages, which are self-mobilizable and use the bacterium to move to another bacteria. And the existence of the hierarchy makes it more complex because now we have different types of alliances. Mobilizable plasmids and conjugative plasmids may ally themselves to move or may be in competition. And this, of course, will change a lot the patterns of horizontal gene transfer. And the situation became even more complicated and interesting 2 years ago, because one came to realize that a lot of what we call bacterial immune systems are not bacterial at all. So we started to analyze these P4 elements. We knew 3 elements until 2 years ago. We now know more than 1,500. Some E. coli genomes have up to 5. And these P4 satellites are very well organized. They have 7 core genes, which are almost always there and almost always in the same order. So they're very easy to study in that respect. And between 2 genes, the integrase and PSU, there is a region which is hypervariable. Within each element, there's typically 1 to 3 genes, but within different elements, you'll find very different genes. So it started to look what kind of genes we found there, because this is just a small element. It's quite surprising to have such an amount of accessory genes. And we came to realize that the majority of the genes that we identify in this region are actually defense systems. That is, they are systems that allow the bacteria to defend itself from the phages. And we find many of the classical systems like restriction and modification, but we also find new systems like, for instance, the Paris one. And the Paris system is particularly interesting in the topic of, in the context of this talk, because it shows you how complex it can be, the antagonistic coevolution between the bacteria and the mobile genetic element. I showed you that bacteria encode restriction and modification systems to protect themselves from phages. And some phages have evolved anti-restriction systems to protect themselves against the restriction and modification systems of the bacteria. Now, these new Paris systems, you could show that it's an anti-restriction system. That is, when the phage infects the cell and carries mechanisms that allow it to block restriction by the cell, this Paris system will protect the bacterial population from the phages. And we now know also actually an anti-restriction system encoded by phages. So these things can go on for in a very complex way. Except that in this case, the Paris system, it's not a bacterial defense system because it's encoded in a satellite. It's a satellite system. So it's the satellite that's protecting itself, and secondarily, it's also protecting the cell from many phages. And this radically changes the way we think about bacterial immunity and radically changes the way we think about the function of these mobile genetic elements in the cell. Because what we've come to realize by our works and the works of many other people is that satellites often encode defense systems, integrative conjugative elements often encode defense systems, prophages encode antiphage systems. And recently, we quantified all this in a Vibrio castor strain, which is an environmental Vibrio strain species, and we realized that almost 3/4 of all the immune systems that we can identify in the species are actually not bacterial because they're encoded within mobile genetic elements. So most bacterial defense systems are not actually bacterial at all. They are within mobile genetic elements. And it's the alliance between the mobile genetic element and the bacteria, because it's in the interest of the mobile genetic element that the bacteria does not die, that leads them to become bacterial defense systems. So how does this change what we think about the processes, the interactions between mobile genetic elements and bacteria, spurring horizontal gene transfer, diversification of bacterial gene repertoires, and therefore bacterial adaptation? Well, it changes quite a lot. We move from a situation where we have the bacteria and the phage or the conjugative plasmid, and then we have all this antagonistic coevolution where the bacteria tries to protect themselves, at least from the phages that will not bring adaptive traits, but of course the bacteria from to start with cannot know. And then you have the phage or the other mobile genetic elements protecting themselves from the immune systems of the bacteria protecting against the resistance of the phages and so on. And from this situation, which was already fairly complicated, we move to another situation where we should see the genome has sort of a playground of mobile genetic elements. These mobile genetic elements have functional interactions among themselves. The satellites will interact with the phages so that they can change to transfer from one cell into the other. The mobilizable plasmids will interact with the conjugative apparatus from the conjugative plasmids to move to another cell. And all these elements, or many of these elements, carry defense systems that will allow them and the bacteria to filter out some types of mobile genetic elements, in particular those that are particularly deleterious, like, for instance, the virulent phages, which will, by killing the bacteria, will actually kill all the mobile genetic elements within it. So why does this change significantly the way we think about bacterial evolution? It is that now that once we observe a defense system in a bacteria, we don't actually know who is defending. Is it the bacteria? Is it the mobile genetic element? And we don't know who's defending from what. Is it from a phage? Is it from a plasmid? Is it from a phage because it's affecting the bacteria, or is it a phage because it's affecting the satellite? The satellite cannot parasitize this phage, and so it tries to keep it out from the bacteria. There is also this multiple antagonistic interactions of exploitation, where some plasmids will exploit other plasmids to move, some satellites will exploit phages to move. And so understanding these interactions becomes critical if you want to understand the impact of a mobile genetic element in driving horizontal gene transfer in bacterial species. And finally, it becomes much less clear who is benefiting from the horizontal gene transfer. Traditionally, one considers that bacteria will acquire genes. These genes may be deleterious, adaptive, or neutral to the bacterium, and these will shape the fate of the bacteria and the fate of the genes within the genome. But now the situation is more complex because we have a lot of intragenomic conflict, and the DNA that may be acquired in a mobile genetic element may be interesting, adaptive for the mobile genetic element, without necessarily being adaptive for the bacteria, or at least not being adaptive for the bacteria in every single circumstance. And so these are three key questions I think will structure a lot of our work and other people's work in the next couple of years, and which will be key for us to understand now that we can access all this microbiome data, and we can look at how things are happening in the environment. It will be key to understand how these interactions take place within the cell, to understand the flux, gene flow in these environments. So to finish, I would like to thank, of course, our collaborators and the people in the lab, Marie Touchon in particular for the work done on E. coli, Olaya Randueles, George Souza, and Mathieu Dickey, who has left the lab for the work on Klebsiella pneumonia, and George Souza, who has been leading the work on the phage satellites, Manuela Arroyo and Charles Kuduzzi, who have been working on the mobilizable plasmids, and all the other people in the lab are somehow working on some type of mobile genetic element that is exploiting or antagonizing another type of mobile genetic element. And I thank you, of course, for your attention, and I'm happy to answer the questions if you have some.
Thank you for a very interesting presentation. And I have a question about kind of the mobilomes. They are very common in inter-species and the cell types, but I wonder there is some kind of the more broadened way to the exchange of the mobilome, like at the genus level or the higher level.
Yeah. Yeah, so we haven't yet worked on that experimentally very much, but we have made experiments between E. coli and Klebsiella pneumonia, so there are different genes, and indeed the capsule does make a difference. So you have much more it's between two different genes actually, horizontal gene transfer if the Klebsiella lack a capsule. So at least for conjugation, which tend to be very broad dose range, you can have conjugation between very differently related bacteria. This remains completely pertinent. For phages, it's harder to say because they do tend to be more narrow host range. So I don't know if we look at inter-species or intergenus comparisons, if you'll find exactly the same patterns. It may be that I would assume that most phages simply will not infect.